Welcome to G Fun Facts Online. In the world around us, and even inside us, a silent war has been raging for billions of years. The combatants are microscopic, but their conflict has shaped all of life as we know it. The predators in this war are the oldest and most successful assassins on the planet. They are not animals, but viruses known as bacteriophages or bacteria eaters, and they are, without question, the most abundant biological entities on Earth. There are an estimated 10 million billion bacteriophages on the planet. That's a 1 followed by 31 zeros. This number is so vast, it exceeds every other organism on Earth combined. Today, we're pulling back the curtain on these unseen architects of our world, from their discovery to their brutal methods of killing, and how these ancient assassins might just hold the key to our future. Our story begins over a century ago, with a tale of two scientists who independently stumbled upon this invisible world. In 1915, an English bacteriologist named Frederick Twort noticed something strange. In his bacterial cultures, mysterious transparent spots were appearing, zones of dead bacteria. He found that whatever was causing this was transmissible, but with limited resources, he documented his strange findings and moved on, leaving the mystery unsolved. 2 years later, in 1917, a passionate, self-taught French-Canadian scientist named Félix Durel made a similar discovery while investigating a dysentery outbreak. He filtered samples from recovering soldiers and applied the filtrate to a culture of dysentery bacteria. Clear plaques or taches appeared, indicating something was systematically destroying the bacteria. Unlike the cautious Twort, Durel was immediately convinced. He declared he had found a virus that infects bacteria, and he gave it a name, Bacteriophage, from the Greek for bacteria devourer. Durel's bold assertion was met with skepticism, even ridicule. The idea of a virus infecting a bacterium was radical, and a bitter dispute erupted over who discovered it first and what the phenomenon even was. Nobel laureate Jules Bordet became a major critic, arguing it was some kind of bacterial enzyme. But Durel was a fervent evangelist. He traveled to India to study why the Ganges River seemed to naturally purify itself of cholera, correctly hypothesizing that phages in the river were killing the cholera bacteria. His clinical trials were often dramatic. In 1919, he famously treated a 12-year-old boy suffering from severe dysentery with a phage preparation. The boy's symptoms reportedly vanished within a day, a success that fueled Durel's conviction and led him to co-found phage therapy centers around the world, from France to the Soviet Republic of Georgia. While his work was eventually overshadowed in the West by antibiotics, the bacteria eaters would soon become indispensable tools in the new field of molecular biology, helping scientists unravel the very nature of the gene. So what do these bacteria eaters actually look like? The most famous of them, the T4 bacteriophage, looks like something straight out of science fiction, a tiny lunar lander with an angular head and spindly legs. But this is just one of a breathtaking variety of forms. The vast majority, about 96%, have a tailed structure. These are divided into three main families. The Myoviridae, like T4, have a complex contractile tail that acts like a powerful syringe. The Siphoviridae have long, flexible, non-contractile tails. And the Podoviridae have short, stubby tails. But beyond the tailed phages, there are filamentous phages that look like long, thin rods, and simple icosahedral phages that are just a protein shell with no tail at all. The core of every phage is its genetic material, DNA or RNA, single or double-stranded, protected by that protein shell, the capsid. The tail is a marvel of nanoengineering. It's a hollow tube for the genetic material to pass through, and at its base, you find the tail fibers. These are the phage's landing gear and its sensory probes. They are responsible for recognizing and latching onto specific receptor molecules on the surface of a target bacterium. This recognition is incredibly specific, like a key fitting into a single, unique lock. This high degree of specificity is why a particular phage can only infect certain species or even specific strains of bacteria, a fact that is critically important for both their ecological role and their therapeutic potential. Once a phage has found its target, it initiates one of two primary strategies. The first is the lytic cycle, a brutal, scorched earth assault favored by so-called virulent phages. It unfolds in 5 swift stages. First, adsorption. The phage's tail fibers lock onto the bacterial surface. Second, penetration. The phage injects its genetic material into the bacterium. In the case of T4, the tail sheath contracts, driving a hollow tube through the cell wall and squirting the DNA inside. The protein shell remains outside like an empty husk. Third, biosynthesis. The phage DNA immediately hijacks the host cell's machinery, reprogramming it to stop its normal functions and start building new phages. The host's ribosomes and metabolic energy are plundered to mass-produce hundreds of copies of phage DNA and proteins. Fourth, maturation. These components then spontaneously self-assemble into new, complete phage particles inside the bacterium. Finally, lysis. The phage produces an enzyme that dissolves the bacterial cell wall from the inside out. The pressure builds until the cell bursts open, releasing a new army of hundreds of phages into the environment. This entire hostile takeover can happen in as little as 20 minutes. But there's another, more subtle strategy, the lysogenic cycle. This is the path of a sleeper agent, used by temperate phages. Instead of immediately killing the host, the phage integrates its DNA directly into the bacterium's chromosome. This integrated phage DNA is called a prophage. The bacterium, now called a lysogen, continues to live and divide normally, and every time it does, it copies the phage's DNA along with its own. The phage propagates in secret, multiplying its genetic blueprint without killing its host, a clever strategy for when potential hosts are scarce. This state of dormancy can last for generations, but it's not permanent. If the host bacterium becomes stressed by UV radiation, chemical exposure, or a lack of nutrients, the prophage can be triggered to induce. It cuts itself out of the host chromosome and enters the destructive lytic cycle, creating a new army of phages that burst out and destroy the cell. This predator-prey relationship has sparked a co-evolutionary arms race that has raged for eons. For every strategy a phage develops, bacteria evolve a counterdefense. Bacteria have developed a sophisticated, multi-layered shield. The first line of defense is to simply prevent the phage from attaching. They can change the locks on their surface, modifying the receptors so phages can no longer dock. Or they can produce thick capsules or slime layers that physically block the phages. If a phage does manage to inject its DNA, some bacteria have molecular scissors called restriction modification systems. These enzymes patrol the cell, chopping up any foreign DNA they find. The bacterium protects its own DNA by marking it with special chemical tags. Another defense is altruistic suicide. Abortive infection systems detect a phage infection and trigger programmed cell death, sacrificing the individual cell to save the rest of the bacterial colony. And then there is the most elegant defense of all, the CRISPR-Cas system. You've probably heard of CRISPR as a gene editing tool, but it's actually a natural bacterial immune system. When a bacterium survives a phage attack, it can snip a small piece of the phage's DNA, called a spacer, and integrate it into its own genome, into a region called the CRISPR array. This array is a library of past infections. If the same type of phage attacks again, the bacterium transcribes the spacer into a guide RNA. This guide molecule then teams up with a Cas protein, the scissors, and patrols the cell. When it finds a match, it binds to the invading phage DNA, and the Cas protein cuts it to pieces, neutralizing the threat. It's an adaptive immune system, and it's passed down through generations. But phages are not passive victims. They evolve just as fast. They can mutate their tail fibers to recognize new receptors. They produce proteins that block the bacteria's restriction enzymes. And in a stunning countermove, some phages have evolved anti-CRISPR proteins that specifically seek out and disable the host's CRISPR-Cas system. This billion-year-old chess match is a primary engine of evolution on our planet. Given their sheer numbers and predatory nature, phages are key players in global ecosystems. In the oceans, they are the masters of the viral shunt. Marine bacteria and algae are vital for converting carbon dioxide into organic matter. Every single day, phages kill up to 20% of all bacteria in the ocean. When they lyse these cells, all the carbon, nitrogen, and other nutrients inside are released back into the water. This shunts a massive amount of organic matter away from larger organisms like fish and keeps it in the microbial food web, where it fuels other microbes. This process profoundly influences global nutrient cycles and even our climate. On land, they are soil sculptors. They use a strategy called kill the winner, where they preferentially infect the most abundant and successful bacterial strains. This prevents any single species from becoming too dominant, which in turn promotes biodiversity and maintains a healthy, resilient soil ecosystem. And they are inside you. Your gut is home to trillions of bacteria, the microbiome, and also an enormous community of phages called the phageome. These phages act as gatekeepers, helping to regulate your gut bacteria, keeping potentially harmful species in check, and maintaining a healthy balance. Phages are also major drivers of bacterial evolution through horizontal gene transfer. They can accidentally pick up genes from one bacterium and shuttle them to another, a process called transduction. This can spread genes for all sorts of things, including virulence factors, the toxins that make bacteria pathogenic, and, most worryingly for us, antibiotic resistance. This rapid sharing of genetic information is a major reason why superbugs can adapt so quickly. This brings us back to Felix Durrell's original idea, using phages to fight infections. In the 1920s and 30s, before antibiotics, phage therapy was a serious hope. Phage therapy centers were established, and companies like Eli Lilly in the US even commercialized phage products for infections. But the results were inconsistent. They didn't fully understand phage biology or the critical need for specificity. Then, in the 1940s, penicillin arrived. Antibiotics were broad-spectrum magic bullets, easy to produce and use. In the West, funding and research dried up, and phage therapy was quickly forgotten, dismissed as folk medicine. It faded into obscurity, though it continued to be developed and practiced in places like the Soviet Union and Poland, isolated behind the Iron Curtain. Today, we're in what many call the post-antibiotic era. The rise of antibiotic-resistant superbugs is a global health crisis, threatening to send us back to a time when a simple cut could be a death sentence. And this has triggered a renaissance for this forgotten cure. The incredible story of Tom Patterson in 2016 brought this into the public eye. He was a professor at UC San Diego, and on a trip to Egypt, he contracted an infection with a bacterium called Acinetobacter baumannii. This particular strain was resistant to every antibiotic doctors threw at it. He fell into a coma and was near death. As a last resort, his wife, Dr. Stephanie Strathdy, an infectious disease specialist herself, began a desperate search for an alternative. She discovered the world of phage therapy and began coordinating with research labs around the world to find phages that could kill her husband's specific infection. A cocktail of phages was assembled and administered intravenously. Within days, Tom Patterson woke up from his coma and went on to make a stunning recovery. His story and others like it have ignited a new wave of research. Phage therapy has huge advantages. Phages are highly specific, targeting only the bad bacteria while leaving your beneficial microbiome unharmed. They work against antibiotic-resistant bacteria because their mechanism is completely different. They even replicate at the site of infection, an auto-dosing medicine that gets stronger as it works. And they are brilliant at destroying biofilms, slimy bacterial fortresses that are almost impenetrable to antibiotics. Many phages produce enzymes that can chew through this protective matrix to get to the bacteria inside. But there are challenges. That specificity, while a benefit, is also a hurdle. It means you need the right phage for the right bug, which requires rapid diagnostics and access to large, well-characterized phage libraries. And just as bacteria evolve resistance to antibiotics, they evolve resistance to phages. The human immune system can also recognize and clear phages, potentially limiting their effectiveness. Finally, regulatory agencies like the FDA are still figuring out how to approve a living, evolving drug. Despite these hurdles, the future of phage applications looks incredibly promising. Scientists are now engineering phages using tools like CRISPR to make them even better killers, broadening their host range or giving them new abilities. Others are focusing on isolating just the lytic enzymes that phages use to kill bacteria, creating so-called enzybiotics that may be easier to standardize as drugs. And combination therapy, using both phages and antibiotics together, has shown amazing results. In some cases, as bacteria evolve resistance to a phage, they lose their resistance to an antibiotic, a phenomenon called an evolutionary trade-off. The applications extend far beyond medicine. A technique called phage display, which uses phages to screen for useful proteins, has been instrumental in developing new drugs and won a Nobel Prize in 2018. Phages are also being used in food safety. The FDA has approved several phage products that can be sprayed on foods like meat and cheese to kill harmful bacteria like Listeria and Salmonella. They can also be used to disinfect surfaces in food processing plants. From a mysterious observation in a petri dish to a central role in modern biology and now a renewed hope for medicine, bacteriophages have had an incredible journey. These unseen architects and ancient assassins are far more than just bacteria killers. They are drivers of evolution, guardians of ecosystems, and a testament to the intricate, dynamic nature of life. As we continue to unlock their secrets, we may find that the story of the most abundant life form on Earth is really just beginning. Thanks for listening to G Fun Facts Online.